Good evening, dear participants. Hope am I audible? And welcome again to this plant genomics and bioinformatics workshop. Today it is a matter of privilege that we have with us today Ensemble outreach team. On behalf of Decode Life, I would like to welcome Dr. Ben and Dr. Elena for today's session. Currently, Ben is working as outreach and training manager for Ensemble and Elena as Ensemble outreach team. So without wasting time, I am handing over session to Benjamin. Over to you, Benjamin. It's all yours now.
Thank you very much for the introduction. Just to introduce myself, my name is Ben and I'm part of the Ensemble outreach team. I'm joined today by my colleague, Elena, and she will also be helping to deliver the session today and tomorrow about using the Ensemble Genome Browser to explore plant genes and genomes. So the session today and tomorrow both have a very particular structure. What we'll do is we'll start off with a presentation. What I'll start today is talking about what the data or the tool is, or what we're beginning to think about as an introduction. And then we're going to move on to a hands-on demo. So the demo is an opportunity for you to begin exploring Ensemble for yourself. So I'll obviously provide a walkthrough showing you the main pages and interfaces of the browser and exploring specific pieces of data that we have in our browser. And you can obviously use that opportunity to follow along if you want to. So in that case, it's probably a good decision to have two different windows open, obviously one with the Zoom where you can follow along what I'm doing, and then maybe a separate window where you can try and follow along for yourself and begin exploring Ensemble. And then at the end of the session, both today and tomorrow, we'll have some time for some exercises. And they're just an opportunity for you to begin exploring Ensemble for yourself. So the exercises go beyond the demo, and they help you to explore all of the different pages that we'd previously seen in the hands-on demo part of the module. There are more exercises than time available. So the idea is that you can spend some time looking through the exercises and picking out one or two that are the most relatable and suitable in terms of your area of research and the species that you're studying. And then you can try having a go at those exercises and obviously move on to some others if you have the time. And you can also finish those exercises in your own time as well. If there are exercises that don't cover your species of interest, then you're definitely not mandated to do the exercises. You can go off course and begin exploring your own species and your own data as well during the exercise session. And Alina and I will be on hand to help answer any questions that you might have. So Alina and I just have two sessions with you today and tomorrow. So what we want to do over the course of those two sessions is to introduce you to the Ensemble Plants resource. We want to teach you about the different types of data that you can get from the Ensemble Plants Genome Browser, and then how to navigate around those pages on the website to get the different data types that you need in your work. We'll also show you where you can go for help and documentation as well. So the first thing that I want to do before I start the introduction is to show you where you need to go for the course materials. So if you open up a new tab in your internet browser, and type the following address, training.ensemble.org into your internet browser, that will take you to the Ensemble training portal. So this is not the Ensemble Genome Browser. This is a separate website that we have set up that contains all of the course materials for the workshops that we deliver around the world. So on this page, you can find a link in the blue bar at the top that says training materials. So if you click there, it will take you to a page that has a calendar of all of our training events. And you can see here, on the 7th of April, we have this event listed here for the Ensemble Plants browser workshop at Decode Life. So this is our workshop, the event that we're having today. So you can click on that link and it will take you through to a page that has all of the information about our course today and tomorrow. So I'm going to copy this link and put it in the chat box for you to use now. So on this page, you can find a link to the presentation slides that we're delivering now. And then you'll also find some other useful links as well. So firstly, we have what we call a living document. If you click on this living document now, you'll find that that link takes you to a Google Doc, which is open for everyone to view and to edit. And this gives you some information about the course. So you can read a little bit of background information about the course on the first few pages. You can find some links to the materials and the feedback survey. And then at the bottom as well, there's also a section for questions and answers. So what we have found in previous workshops is that the chat box in Zoom can sometimes become quite busy and complicated if there's lots of people asking questions all at the same time. So this is a good place to ask questions for us to answer. So the idea is that anyone who's attending the course can come here, and then you can enter your name by editing the document, and then you can just paste in your question here. You can type the question, you can add screenshots, you can add links, and then Alina or I will be able to come back here and enter our answer underneath your question. What this means is that everyone can see the questions and the answers, and then this becomes like a knowledge bank where you can come back to and refer back to everyone's questions and the answers that Alina and I have provided. So I can see lots of you have already entered the document. So yeah, please feel free to use all of the information here, and Alina and I will keep our eye on this document to see if there are any questions that come up. Going back to the training pages with the training materials, at the very bottom of this page, you can see a section called demos and exercises. So there's 3 different modules here that you can see for our workshop today. There's one called Species and Genome Assemblies, one called Exploring Genomic Regions, and a final one called Genes and Transcripts. So if we just take a look at this first module called called Species and Genome Assemblies, you can see that if you click on the title of the module, you're given 2 different options, one for viewing the demo. So if you click on the panel there, it will open up the walkthrough that we're going to be doing later on in this workshop today. So you can find a description of the different pages and the screenshots here. And then at the bottom, you can see that we have this section called Species and Genome Assembly Exercises. So here you can see that we have exercises that you can do at the very end of the module. So the exercises are written here, and you can also expand these panels to view the model answers. And also we have videos showing you how we got those answers as well. So the exercises are not a test. You're not supposed to submit the answers anywhere, or you're not going to be graded or marked on them. The exercises are designed to help you explore Ensemble for yourself and continue learning. So if you get stuck, you can definitely ask Alina and me to help you, but you can also look at the answers here to help you if you do get stuck on something. Good. So hopefully you've all found the materials and they're all available for you. I'm now going to go back to the presentation and we'll begin our introduction to the Ensemble Plants genome browser. So genome browsers are very important. You might have heard of Ensemble before, and you might have heard of other genome browsers as well, such as UCSC and the NCBI genome browser as well. They're all obviously very similar in the way that they present genome sequences and genome annotation. The first genome actually to have the first organism actually to have its genome sequenced was a bacteriophage back in 1977. Fred Sanger and his team here in Cambridge in the UK sequenced a bacteriophage that had a genome that was just 5 kilobases in size. So by today's standards, that's obviously really small, a very small size for a genome. But that technology laid the foundations for more and more complex organisms to have their genome sequenced until in the early 2000s, we had the publication of the first draft of the human genome sequence, which is 3 gigabases in size, 3 billion base pairs. Even since then, we have the advent of next generation sequencing technologies, which means that genome sequences are being sequenced at a much faster rate for a much smaller cost. And what that means is that there are lots of large-scale, worldwide collaborative projects that are generating genome assemblies for lots of different species all around the world. So as an example, we have the Darwin Tree of Life project, which is aiming to sequence the genome of every eukaryote in the British Isles. But then we also have projects such as the Vertebrate Genome Project, which is aiming to sequence the 60,000 vertebrate species that are existing around the world. So the idea that I'm trying to impress upon you is that genome sequences are becoming very, very abundant. They're being generated almost every hour, every day. And so it's the job of the genome browsers to take these genome sequences and then annotate them and then to present them to scientists all around the world to access them and use them in their research. So when a species has its genome sequenced, what we generally see out of the end of that sequencing run is just a string of letters, A's, T's, G's, and C's. So without any context or without any annotation, this doesn't mean anything to anyone. Even a scientist looking at this can't really see what's going on here. So what Ensemble does is we take these genome sequences and we annotate them with lots of different data types. So we annotate the gene and the transcript models. We annotate variation data. We perform some comparative genomics data, some comparative genomics analyses. And we also add regulatory data for promoters and enhancers as well. So Ensemble is not a sequencing project. We rely on these different projects and research groups around the world to sequence the genomes of different species and then to deposit those genomes into publicly available archives. So once those genome sequences have been made publicly available, Ensemble can take those genome assemblies, the genome sequences, and annotate them with all of the data that we annotate genomes with, and then make them available through our website and our other databases. So as you can see here, this is just a diagram or a cartoon to try and explain to you how it works. So we take these publicly available genome sequences from the archives where people have deposited them, and then we annotate them by mapping genomic features using a range of evidence and underlying data. On top of that, Ensemble is an added value resource, meaning that we also bring together a wide range of information from other databases as well. So we draw in information about genetic variants, clinical associations, literature citations, and we bring those all together and we present them through one web page, like one website. So Ensemble has, as I've sort of already alluded to, Ensemble has 4 main types of data. So firstly, we have the main type of data is the genomic assemblies themselves. So this is the genome sequences that are produced by different groups and research collaborations around the world. They're deposited into publicly available archives, and then Ensemble takes them and performs a gene annotation process. So we have an automated gene annotation pipeline that predicts the position and the structure of the genes and their transcripts. The second type of data is the variation data. So this is both small and large scale sequence variants, and we also include phenotype associations and allele frequencies and literature citations where those are available as well. The third type of data is the comparative genomics. So this is where we compare the genes, the gene models and the genome sequences from one species to another to look at things like homology relationships and conserved regions. And then the fourth type of data is the regulation data. So this is where we take epigenetic data such as ChIP-seq data and DNA methylation data to predict the existence of regulatory features such as promoters and enhancers. These are features that regulate gene expression. So when we're thinking about Ensemble Plants, Ensemble Plants has a number of features to draw these four main branches of data together. So we have gene builds for 119 different plant species now in our Genome browser. One of the data types that I mentioned as well is the comparative genomics. So we have alignments, gene trees, and homology predictions for all of our Ensemble plants. And we also have the display of variation data for a number of different plant species that have variation data available for them. And we also have a tool called the Variant Effect Predictor. You can see the acronym here is VEP, V-E-P. And this is a tool that allows you to begin analyzing your own variation data sets. So if you've performed some sequencing yourself and you've identified variants in your own work, you can use the Variant Effect Predictor tool that we have here for analyzing the consequences of the variants that you've identified. For plants, it's important to notice here that we don't provide any regulatory data for plants. So as I mentioned on the previous slide, we have this annotation of regulatory features, but this is only available for human and mouse at the moment. So we don't have any regulatory feature annotation for plant species because the data is not available for us to perform that annotation. As well as this data that we have for the plant species, you can also visualize your own data in the Ensemble Genome browser. So you can display your own data alongside the publicly available Ensemble data as well. So it's a way for you to visualize your data and to compare it against the publicly available data that we have in the Ensemble Genome browser. And all of this data and all of these tools are available through the Ensemble website, and we're going to show you that later on in the demo today. But you can also begin thinking about exporting this data on a larger scale. So the website is really good for looking at individual genes or individual features at a time. But obviously, sometimes you might have a larger scale analysis where you want to look at a number of different genes all at the same time. So in that case, we have some tools that you can consider using if you are performing this larger scale work. So you can use the Biomart data export tool through the Ensemble Plants website. And you can also access the data programmatically through the REST API as well. The best thing about this is that the Ensemble data is completely open source. So all of the data, everything that you see in Ensemble is completely free for you to take and use and then analyze and publish within your work as well. So Ensemble as a project is dedicated to vertebrate species. So those are species with a notochord. And you can see here a representation of the different species that we have in Ensemble. So we obviously have a large number of, we have the great apes. So we have human, orangutan, gorilla, chimpanzee, and bonobo. And then we have a large number of other primates, and then also placental mammals, thinking about conservationally and agriculturally important species as well. And then we have a large number of other marsupials and monotremes. So trying to study mammalian evolution is obviously very important. So we have the three different groups of mammals. And then we also have a large number of birds, reptiles, fish, and amphibians as well. Just highlighted on this tree, you can also see that we have three non-vertebrate species in Ensemble. So we have C. elegans, the nematode worm. We have Drosophila melanogaster, the fruit fly. And we also have the baker's yeast, Saccharomyces cerevisiae as well. So these are three non-vertebrate species that we include in Ensemble because they're very important model organisms. And they also form a lot of the basis and anchoring of our gene trees as well. So obviously what we are covering today is Ensemble Plants. So what we also have is a sister project called Ensemble Genomes. So if you're studying a non-vertebrate species, we have a sister project that presents the genome sequence and annotation data for non-vertebrate species. So Ensemble Genomes is the website, www.ensemblegenomes.org. And we have 5 separate pages for the different taxonomic groups that we have in Ensemble Genomes. So we have Ensemble Bacteria, Ensemble Protists, Ensemble Fungi, Ensemble Metazoa, and importantly for us today, Ensemble Plants, which we're going to explore in a lot more detail. So when you're using Ensemble and Ensemble Genomes, it's very important to remember that the Ensemble and the Ensemble Genomes resources work in a release cycle. So new genomes are always being sequenced, new data is always being published and put into different databases. So Ensemble releases a new version every 3 to 4 months. And this means that we can incorporate the latest data into the Ensemble Genome Browser as well. So what happens is that we look for new species that have had their genome sequenced or had an updated annotation data set. We update our underlying software and we reperform all of the different comparative analyses and update the different data sets. And then we package that all up and we release it as a new version of Ensemble. So at the moment, we're in Ensemble 105, which is the current version of Ensemble. But later on in April, we're going to release Ensemble 106, which will be the latest version of Ensemble that includes the most up-to-date data. The most useful thing to remember is that as well as having the current site that has the current version of Ensemble, we also make the archives of Ensemble available as well. So if you were looking at Ensemble last year, maybe you were using Ensemble 104 last year, you can actually visit the Ensemble 104 archive and view the data as it appeared at that time. So what that means is that if you retrieve some data from Ensemble and then perform some work and then come back to Ensemble later on, you can always go back to the data as you saw it at that time. So you can check your analyses and see the data as it would have been when you first downloaded the data set. The final thing that we have recently published is a separate website called Ensemble Rapid Release. So as I've mentioned, this release cycle takes quite a long time to integrate all of the different data sets every 3 to 4 months. And at the moment, more data is being produced. The data is being produced at a rate that sort of is quicker than this release cycle. So what we've developed is a site called Ensemble Rapid Release, which is released every 2 weeks, and it contains a minimal amount of gene annotation data for newly sequenced genome species. So we have the genome sequences with the gene annotation available, and then the BLAST tool, but there's no biomart or comparative genomics analyses or variation data available. So we call it like a light version of Ensemble, meaning that it's sort of it just contains the core data sets that can allow people to begin exploring the different species that have been recently sequenced as part of the different sequencing projects. So when you are looking in Ensemble, as I mentioned, one of the things that, well, I've used the word genome sequences and genome assemblies quite interchangeably. And this is because in Ensemble, we actually present what we call the genome assemblies. So at the moment, there's no technology that can sequence a whole chromosome in one go. So what we actually have to do is fragment the chromosomes into short reads, and we sequence those short fragments, as like you can see here, to produce our sequence reads. Then we match up our overlaps. So we take the sequences of our reads, we match up their overlaps to see where they align, and then we infer the sequence from the matched up overlap. So you can see here, for example, we have these 4 different reads that all suggest that at this position there is a T. So in our genome sequence, we have a T. And where we have all of these matched up sequence reads, we call this sequence a contig. So contig is short for contiguous sequence. So that just means that all of these reads are forming one inferred sequence from these fragments. So obviously, once we have these contigs, we need to build them up and up to put them back together to form the whole chromosome. So when I'm thinking about genome assemblies, I often like to think of it in the context of a puzzle. So when you're looking at a puzzle with maybe 500 pieces, you know that you have an overall picture that you're trying to put together all the different pieces to see the final picture, but you have them all obviously separated. So the idea of the puzzle, as is the idea of the genome assembly, is that you're putting these pieces back together to obviously get the overall final picture that you're looking to see. And that's exactly the same principle with the genome assembly. So this slide is slightly out of date now with the advancement of the different sequencing technologies. But when the human genome was first sequenced, what we actually had to do was we took these individual reads and we cloned them into bacterial artificial chromosomes called BACs, bacterial artificial chromosomes. And then we sequenced those clones, and that's called the tile path. So you can see here, these 4 different blocks here, these are all individual clones that together form the tile path. Then the overlaps of these individual clones within the tile path are trimmed. So this overlapping region is trimmed to give us our contigs. So you can see here, this is contig 1, for example, that comes from this back clone here. And this is another contig that comes from this other back clone here. So this is how the contigs are represented in Ensemble. They're represented as these blue lines that are made up of light and dark alternating blue colors. So a run of contigs with no gaps is called a scaffold. So there's a couple of questions that have come in here. It says, so on the chat box, this PJ has said, could you please explain contig? And then Sweeta, what is the difference between contig and scaffold? So hopefully I'm answering your questions as I describe this here. So to answer Sweeta's question, a run of contigs with no gaps is a scaffold. So obviously this is a simplified version with just a small number of contigs, but here you can see that this would be one contig here, that's one scaffold that's made up of 2 separate contigs. So a scaffold is a run of contigs together without any gaps. And then also, PJ, I hope I've answered your question here, where I've explained the contig sequence that comes from these back clones. And then finally, we take these scaffolds and we place them onto genetic maps to work out their position relative to one another and their orientation to build our final chromosomal sequence. So this is what we represent. This is not work that's done by Ensemble. This is work done by these research groups that sequence the genomes. And then this is the sorts of data that they deposit into the publicly available archives that Ensemble can see and access. So it's very important to remember that when you are looking at a genome assembly in Ensemble, as I sort of mentioned, it's a puzzle that we're putting back together. And obviously, when you're working at a molecular level, then there might be pieces that are missing or difficult to find where they're actually located. So the genome assembly that we actually have is a representation of the genome that might contain errors or gaps. So again, for an analogy, I think of this in terms of like the world and the different types of maps that we have of our world. So the genome itself might be the world itself. And obviously we have lots of different maps that represent the world. Obviously today with our technologies, we have Google Maps, which is a very complete representation of the planet. But obviously back in the 16th and 17th centuries, when people were exploring and there wasn't the technology available, we did obviously have maps, but they had lots of gaps and errors and mistakes. And that's exactly the same with genome assemblies. So genome assemblies that were produced a few years ago might have some gaps and errors where the technology wasn't able to create a perfect picture across the whole genome. But now, obviously, the technology is improving, so we have new versions of genome assemblies that are improvements on ones that have come before. So what I want to do now is we're going to explore the Ensemble Plants on a genome browser. So we're going to look at the different species that we have available, and we're also going to explore the genomic location. So we're going to look at a region of the wheat genome, So wheat, as we know, is a hexaploid organism. So it has three copies of so it's a polyploid genome. It's got three copies of the chromosomes, 3 copies of the diploid chromosomes. So we're going to look at chromosome 3D in wheat, and we're going to look at this genomic location using these coordinates here. So what I will do is I'm going to copy these coordinates and I'm going to put them in the chat box, and we can copy and paste them together at the right time in the demo. So what we can do, if you want to follow along with me in the demonstration, we need to open up a tab in our browser and go to plants. Let's start from the beginning. So we can go to www.ensemblegenomes.org. So this is our sister project that has all of the different genome assemblies for the non-vertebrate species. So you can see a link to our coronavirus genome browser, our SARS-CoV-2 genome browser. We also have a link to the Ensemble rapid release site, as I mentioned before. And then we also have links to our 5 different division pages. So we've got the plants, metazoa, protists, fungi, and bacteria as well. Now, obviously, I know that we're looking at plants today, but obviously it's important to study plants sometimes in the context of plant pathogens, so looking at fungi and bacteria, and sometimes even metazoa if you're looking at aphids and those sorts of things. So you can always come to these pages and go to the specific pages that have information about the species that you're interested in. Obviously, for our demonstration today, we're interested in Ensemble Plants. So we can click on the link here, and it will take us to the Ensemble Plants genome browser. So what you can see in the address bar is that we've navigated straight to plants.ensemble.org. So this is actually the web address of the Plants genome browser. So if you want to come directly here, you can just type that address straight into your genome browser, plants.ensemble.org. And I'm going to put that. I'm going to copy that and put that in the chat box so that you can also navigate here if you're trying to follow along as well. So on the homepage, you can see lots of information, and I'm going to try and break it down for you so you can see everything that's there. So at the very top, we have this green navigation bar. So this will appear at the top of every single page in the Ensemble Genome browser. And so you can always use the links here to continue searching or to navigate to another page. So the logo is actually a link back to the homepage. So if you're navigating and exploring and you want to come back here and perform a new search or look for something different, you can click on the logo to come back to the homepage. Then we have a link to some different tools. So we've got our hammer tool, our blast tool, and the Biomart data export tool. We also have a link that's called tools that has links to our other tools. So if you click there, you can see that we have links to the variant effect predictor, the hammer and blast tools, as well as the assembly converter and the ID history converter as well. So we won't have time to look at all of these tools individually, but you can come here and explore them in your own time and see if there are any tools that can help you in your work. If you come back to the top left-hand corner, you can click on Ensemble Plants, and that will bring you back to the home page. And you can also see here we have a link for downloads. So if you want to download the genome sequences or the sequence files for a particular species, you can click on the downloads link to access the files themselves. And you can also find the help and documentation in the green bar at the top as well. Underneath the green bar, we have this search box. So this is the main portal into the rest of Ensemble. So in this search box, you can search for genomic coordinates, you can search for gene names, you can search for gene IDs, you can search for variant IDs. So anything that you really can think of, you can search in this box here, and we're going to do that in just a moment. Underneath the search box, we have information about the different species that are in Ensemble. So we have this quick links for some commonly accessed species. So we have Arabidopsis, Thaliana, we've got the rice Orza sativa japonica group, we've got the wheat Triticum aestivum, and then we also have Hordeum vulgare, barley, Z maize, and also this Physcomitrum patens as well. This is not all of the species that we have available. These are just quick links for commonly accessed species. If you want to see all of the species, you can either click on this drop-down menu here and pick your species of interest, or you can click on this link that says view full list of all species, which we're going to do in just one moment. Over on the right-hand side, you can see some information about the different species as well. So we've got a bit of a description about some of the recent data that we added for wheat. And you can also find links to our archive sites as well. So as I mentioned, we have archives of Ensemble and the Ensemble Genomes pages. So you can access the archives through the links here. Then at the bottom, we have information about the different releases. So you can see here, we're currently in Plants release 52, which correlates with Ensemble 105. So both Ensemble and Ensemble Genomes have the same release cycle, but because Ensemble is an older project, the release number is higher. So at the moment, we're in Ensemble 105 and Ensemble Plants 52. And then when Ensemble 106 is released, we will release Ensemble Plants 53 at the same time. So they go at the same time, but the numbers are just different because the one project is older. Good. So the first thing that you might like to do from the homepage is to search for the species that you're studying, your species of interest. So you can click on this link here that says view full list of all species.
Hello, sir.
Hello. Is there a question?
Sir, yes, sir. I have one question, sir.
Of course.
Sir, actually, I'm working on a chickpea, a legume plant, legume crop. Sir, as you mentioned, most of the plants information is available in Ensemble plant, but there is no information of chickpea. Oh,
okay.
Actually,
yeah, so
actually, I have around 50 protein sequences of chickpea plant.
Yeah.
And I want to identify the gene information, all of these, and promoter in a single click. But I don't know the method that how you can get information and how you can identify all the gene information of genes and promoter sequences in a single go.
So that's a very good question. So sorry, I didn't mean to cut you off there. Did you have anything else you wanted to say?
Sir, actually, I have 50 protein sequences of chickpea plant.
Yeah.
And I want to get information of all these genes that are responsible for coding these proteins and analysis of promoter analysis of all these genes.
Yeah.
I am unable to get information about the gene and promoter sequences.
Yeah, so it's a good question. So obviously there are lots of different species, plant species that have had their genome sequenced. And we work as quickly as possible to put as many species into Ensemble Plants as possible. So the species that we have in Ensemble and in Ensemble Plants obviously depends upon the number of species that we have in Ensemble Plants depends upon the quality of the genome assembly. So what we'll do is we'll assess the quality of the genome sequences and how complete the genomes are. And we'll also look at the demand within the community for incorporating particular species into Ensemble Plants. So at the moment, I think you're correct, is that we don't have the chickpea genome sequences in Ensemble. But I think actually, well, I don't know the timeline for this, but I do know that we've been discussing chickpea with the Ensemble Plants team. And I think that we do have plans to incorporate chickpea into Ensemble soon, but I'm not sure of the timeline. I can look into this and get back to you at the end of the workshop today. So when we are in Ensemble Plants, so yeah, going back to the demonstration. So if you click on view full list of all species, that will take you here to a table that has all of the species that we have in Ensemble. So you can obviously scroll down and begin to see all of the different plant species that we have available. But you can also use the filter in the top right-hand corner here to search for your species of interest. So you can see here, for example, if you want to search for wheat, you can see that you can search for Triticum. You can begin typing the name of the species, Triticum isovam, and now you can see all of the different Triticum isovam genome assemblies that we have. So interestingly, you can see that for wheat, we have the Triticum isovam reference genome assembly here that's produced by the IWGSC. So this is the name of the genome assembly, and this is the people who produced it. But then we also have a number of different wheat cultivars as well. So depending on the species that you're looking at, you might have just one reference genome assembly. But for some species, we have different strains as well. So for wheat, we have these different cultivars that we can also view as well. So if you want to find out more information about a species, you can look in the table. So you can see that the wheat genome assembly here has got an accession number. So this is the unique ID that refers to this the genome assembly that we have presented in Ensemble that's produced by IWGSC. And you can see that we have a variation data set available for wheat. We also have a regulation database, whole genome alignments, and peptide comparer as well, so comparative genomics analyses. If you want to find out more information about a species, you can click on the species name in the table. So you can click on Triticum isovam, and that takes you to a species specific homepage. So from here, you can find a search bar to search specifically for the bred wheat. You can search for gene names or genomic coordinates from here. You can read a little bit of information about wheat as well. And then at the bottom, you can see these 5 different boxes that give you information about the genome assembly itself, the gene annotation data, the comparative genomics data, the variation data, and the regulation data. So we're going to explore the different data types a bit more with Alina tomorrow. But what we can see here is this section called Genome Assembly. So you can see that the name of the genome assembly is IWGSC. And if you want to find out more information, you can click on the link here that says more information and statistics. So this will take you to a page that has information about the genome assembly for this species. So there's some background information about who produced the genome assembly and when. So you can see here, the assembly was produced by IWGSC, and you've got the link for the accession number for this genome sequence. And then you also have information about the annotation. So you can see who produced the gene annotation and where it's been, how it was being produced as well, and where any other data sets have been drawn from as well. You can also see a table that shows you some summary statistics for the genome. So the number of genes that have been annotated across the whole genome, both coding and non-coding, for example. Good. Okay. So what I'm going to do now is I'm just going to click back onto the name of the species in the green bar at the top. And that will take me back to the species specific homepage. So what I want to do now is explore a specific region of the wheat genome. So I'm going to use the coordinates that we searched, that I put into the chat box earlier. So I'm going to paste those coordinates into the search box you can see here. So when we are searching for genomic coordinates, we have to have a very specific format that we are using. So we always start with the chromosome number. In this case, we're looking at chromosome 3D. Then we have a colon. Then we have our start coordinate, which in this case is 47669300. Then we have a hyphen, so just this small dash here. And then we have our end coordinates, which is 4771100. So we always have chromosome number, colon, start, hyphen, end. So this is the format for our genomic coordinates. So when you've entered this into the search box, you can click go. And this takes you to what we call the region in detail page. So just as the page loads, I'm going to just explain some of the different features on the page that you can see. So you can see in the green bar at the top, we've opened up this link called location. So what you're going to notice is that when we are searching for different features and looking at different things, we're going to open up different types of tabs. So at the moment, we have a tab here for the genomic location. But if you now click on a gene or search for a gene, then you're going to open up a gene tab. So we have different types of tab for different types of features. So obviously we searched for a genomic location. So we've now opened up a location tab in the green bar at the top. Underneath the tab, you can see that we have this menu over on the left-hand side. So we've landed on a page called the region in detail. This is the page that we're viewing at the moment. But you can also see some of the other links in the menu on the left-hand side that will help you look for data associated with this region. So you could look at some comparative genomics analyses. So you could look at alignments between different species. And you can even look at the polyploid view here, which is available for wheat, for comparing across the different hexaploid chromosomes within wheat. Now that the page is loaded, underneath the menu, you can see these 5 green buttons over on the left-hand side. And these are really important because they help you configure and manipulate the data that you see on the page. So the first button is called Configure this page. We won't click on this now, but we're going to come back here later because what this allows us to do is to add more data to this view. So by default, we just show you a small amount of core data, but there's lots of different data that you can add to this view. And we're going to see that how we can do that later on in this demo. The button underneath is called Custom Tracks. So this allows us to visualize our own data sets within Ensemble. So if you have your own data set that you want to visualize, if you have your own data set that's aligned to the wheat reference genome, then you can click on Custom Tracks here and upload your data to visualize against the wheat data. The next button underneath is called Export Data, and I'm going to click on that now. When you click on Export Data, you should have a pop-up window that opens. So I'm just waiting for my I'm just waiting for this page to load. There's a question that's just, while I'm waiting for this page to load, there's a question that's coming from Rishika. It says, this chromosome 3D for a particular species means what? So maybe I can open up a new tab just to answer this question for you. So if I go to plants.ensemble.org, I'm going to go just back to, I've opened up a new tab and I'm going to go back to the home page, although everything's working very slowly at the moment. If we go to the wheat homepage, you can actually click on the link here called View Karyotype. So you can see that this is the karyotype of the wheat genome. So you can see that because wheat, the bread wheat triticum isoform is formed by two separate hybridization events of diploid genomes. So over the course of for many hundreds of thousands of years, obviously wheat has been bred, and in that breeding process, there's been these hybridization events. So initially we had these two species that were both diploid, and then they obviously hybridized to form a single tetraploid genome, and then we had a tetraploid genome that was then hybridized with another diploid genome to produce our hexaploid genome. So we have 6 different chromosomes. So we have 3 copies. So we have 2 copies of 3 separate chromosomes. So you can see here we have 1A, 1B, and 1D. So this is the 3 different copies of chromosome 1. Then we have 2A, 2B, and 2D. So these are the 3 separate copies of chromosome 2. And then we have the same for chromosome 3, chromosome 4, chromosome 5, chromosome 6, and chromosome 7. So these are the 7 different wheat chromosomes that have 3 copies each. So I hope that makes sense to you. Going back to our demo, we can see that this page is now loaded. So what I had done was I clicked on the green export data button. So now when this pop-up page loads, you can see that we are able to export the data from this region. So the first option here that we have available is the output option. So we can choose the different output formats that are available to us. So if we want to download the sequence, we can choose FASTA. But if we want to download the features from this region, such as the genes or the other features, we can choose these other format types as well. So if I want to choose sequence, I can click FASTA sequence. I've got the coordinates already pasted into the box here, and then I can click next. And then from here, I can either download the file of the FASTA text file, or I can click on HTML, and that will show me the sequence of this region as well. So obviously I can then take this FASTA sequence and I can upload it into other tools, so I can continue my analysis. I can do some primer designing analyses. I can put it into primer designing tools, all those sorts of things. So that's the way to export the sequence of this genomic region or any other features within this region. Good. So when you're finished with the downloading, you can click on the tick in the top right-hand corner, and that will get rid of the pop-up window and just come back to this main page. The other options over on the left-hand side are to share this page or to bookmark the page as well. So that's obviously helping you to keep this page for later on when you come back to Ensemble. Further down, so on the main part of the page, you can see that we have these 3 separate images. So you can see at the top, we have what we call the chromosomal overview. So you can actually see that this is just a depiction of the whole chromosome. So you've got the chromosome here with the centromere, and you've got this red box. Although at the moment, it just looks like a line because we're quite zoomed out. But obviously, this is our region of interest, this red box. And we're beginning to, as we go down the page, we're going to zoom more and more into our region of interest, this red box. So when we look at the image underneath, you can see that this is a 500 kilobase view around our region of interest. So you've got the scale at the top here. You can see 500 kilobases. And then we have this red box, which is our region of interest. So this is the region that we originally searched for. And we can see the upstream and downstream sequence as well. So the important thing to notice is that the data is now arranged into what we call tracks. So we have what we call a contigs track here. And the contigs track is what you can see, this blue bar. So as we discussed in the presentation before, the contigs are the puzzle pieces that form the whole genome assembly. So this is what we actually see here, this blue bar that represents the double-stranded DNA sequence itself. So this is the reference genome sequence here, the blue bar, and the data in all of the other tracks underneath is aligned to the reference genome sequence. there's a couple of questions that have come in on the chat box. So I'm just going to finish the demo section that I'm doing at the moment, and then I'm going to come back to any unanswered questions at the end as well. So yeah, please, I can see the questions, and we'll revisit them at the end. So yeah, don't worry, I haven't forgotten them. So you can see that the data in the other tracks is underneath the contigs. So here we have, for example, what we call the genes track. So you can see the genes are represented as these colored blocks, and you can see the ID of the gene underneath as well. So you can see here, this block here is this gene with this ID, and you can see this is a separate gene here, and this is the ID of the gene underneath. The color of the block represents the biotype, so you can see all of these red colored blocks, these are protein coding genes. If we scroll all the way to the bottom, we can see our final image, and this is what we call the region in detail view. So the red box that we saw here, this is our region of interest. So what we see in this final image, this is our red box. So this is our start coordinate that we originally searched for, and this is the end coordinate that we originally searched for as well. So this is the region that we're interested in. And again, we can see the data is arranged into these different tracks. So we've got the contigs track, as we said again, representing the double-stranded DNA sequence. And then we have the data in these other tracks. So we have a genes track here. And you can actually see in this view that the genes are no longer represented as just individual blocks, but they're represented as the transcripts that are made up of different exons and introns structures. So Alina is going to talk about this in more detail tomorrow. But if you're interested on a gene, in a gene of interest, if you're interested in a gene in this region, you can click on it and get more information. So this is where you can click on all of these different features and get more information by clicking on them in the view. Good. So what I want to do now is show you how you can add data to this default view that's appearing here. And you can do that by clicking on the green Configure this page button over on the left-hand side. So if you click on Configure this page, you can see that in the pop-up window, we are shown all of the, well, in the first view, we're shown all of the active tracks. So these are all of the tracks that are shown and turned on by default in this view. But over on the left-hand side, in this menu, you can see all of these different subcategories that show us the different tracks that have different data types to display in them. So for example, we have mRNA and protein alignment tracks. We have comparative genomics tracks, and we also have tracks showing us variation data as well. So for example, if you're interested in viewing the wheat variation data, you can click on variation in the menu on the left. And you can see all of the variation data tracks that are available. So you can see here we have this one track that's already turned on by default, and we can tell that it's on because we have this icon in the box here. So that means it's currently turned on, but all of these other tracks are currently turned off. So for example, if you wanted to specifically see variants from the serials DB, so this is the serials database variants, you can turn a track on by clicking on the box, and then in the pop-up window, selecting the format. So I'll select normal in this case. If you're not sure what a track is or what data is going to be displayed, you can click on the information icon and it will just show you, it will give you a bit more information about what the data in that track is going to show. As well as searching in the menu on the left-hand side, you can also look for tracks using the search function in the top left-hand corner. So if I want to turn on a repeat feature track, I can just type repeat, and then I can find all of the different repeat features tracks that are available. So I can turn on a track that shows me all of the repeats in this region. So when you're happy, with your with the tracks that you've added, you can save and close by clicking on the tick in the top right-hand corner. And you'll see at the bottom here that this page is now reloading. So we can see now that we have added this track that's called variant serials DB. So we can see any variants in this region from this specific serials DB database. And we can also see this track called repeats. So these are the repeats, which you can click on and find more information about. So obviously I'm not going to be able to show you every single track that's available. The idea is that you're during the exercises, and then when you're using Ensemble in the future, it's best to look at your species of interest, explore the different tracks that are available, and try and think about which tracks are going to show you the data that you need in your work and in your analyses. So the final things that I want to show you that you can do is that you can actually manipulate the tracks from this view itself. So if you click on a track name, like we'll click on this repeat track here. If you click on the name, you are given some options. So you can read a little bit of information about the track, you can share the track, or you can turn the track off by clicking on the cross. So if you no longer want to see this track, you can click on this cross, and that will remove the track from your view. You can also move the tracks relative to one another. So if you hover your mouse so that it goes over this colored bar on the left-hand side and turns into the arrow icon, you can click and drag your tracks to configure them how you want. So you can obviously view these different data types more closely, like aligned to one another. So you can see them in close proximity to one another. What you can also do is you can use these two options up in the top right-hand corner to continue searching and exploring. So you have this dotted box option and the double-headed arrow option. So if you click on the dotted box, it means that when you click and drag with your mouse, like this, when you let go of your mouse, you're given an option to either mark the region or to jump to a region. So if you click jump to region, this whole page will reload, focused around that new box you just drew. So obviously this is a way to begin zooming in and looking at specific regions that you're interested in. If you change to a double-headed arrow, if you click on the option up here, it means that when you click with your mouse, instead of drawing a box, you can now click and drag the genome. So you can begin moving upstream and downstream, exploring the different features in that genomic region. The final things that you can do are in the top left-hand corner here. So you can export this image. So once you've found a particular area of genomic region that you're interested in, you can export the image, obviously for publication or presentation or poster. And then you can also use the buttons here to either reset the track configuration, so to go back to default, turn off all the tracks that you had originally turned on, and then you can also reset the track order as well. So this helps you go back to default by clicking these two options here in the top left-hand corner. So that brings me to the end of the demo. What I'd like to do is just quickly look in the chat box to see if there are any questions, and then I'm going to set you with some exercises to do in the last part of the session today. So I can see that there's a couple of questions that have come in. I think Alina has answered a question about contigs, and she's also answered a question about getting whole genome data. I think there's an outstanding question here from Dr. Sava Mangala, and it says, how best can I explore Ensemble Plants for my RNA-seq data? So I think the best thing, I haven't seen your data set, but I think the best thing for you to explore your RNA-seq data set in Ensemble is to click on this button here on the left-hand side called Custom Tracks. And what you can then do from this view is you can upload your own RNA-seq data set to explore, to visualize in the Ensemble Genome Browser. So I'm guessing if you have an RNA-seq data set, it's probably a BAM file that you have. if not, then we do support other file types as well. So if you have a BAM file with your RNA-seq data set, you can upload them using the box here. So you'd obviously give your track a name like this, and then you would put your data in this box. So you can either paste your data directly in this box if it's just a small set of data, or you can upload a file like using the option here. Or if you have a really large RNA-seq data set, then you can place your data sets, your files, into a public directory like an FTP server from your institute, and then you can put the web address here. So obviously something like this. So you'd obviously have to place your file in a public directory and then link to it by giving the URL here. What that means is that, so when you put your data into the Ensemble Genome browser here, that's not uploading it so that everyone around the world can see it. That's just uploading it so that you can visualize your data alongside the publicly available data that we have in Ensemble. So in that way, you can begin comparing your RNA-seq data set to the gene models and the other data that we have presented in Ensemble as well. Good. So there's another question that's just come in from Dr. Foram Vaidya. So it says, how can we explore Ensemble for mapping in transcriptomics analyses? So I think this is the exact same question that we just had. So transcriptomic analysis is probably going to be an RNA-seq data set that you have from different plant tissues. So the best thing to do is to look for the reference genome assembly for your species of interest in Ensemble, and then upload your data set using the custom tracks option. Another question has come in. Can we get new gene IDs from de novo assembled novel genes? The answer is no. So Ensemble just has obviously the reference genome sequences and the annotation that we present in Ensemble. So Ensemble is not a tool for performing your own genome assembly or your own gene annotation. You can visualize your data, but you can't do any annotation using the Ensemble platform. You have to use other tools for that. And then another question that's come in from Sweta. It says, can I submit my GBS data set? So maybe you can paste back into the chat box. I'm not sure what GBS means. Maybe it's something that I'm not aware of, but if you could explain what data set you have, then I can maybe see whether it's possible to work with that in Ensemble. Atul says, where can we get the GTF file of the genome required for RNA-seq analysis? So I can show you this in the demo. So if we go back to the Ensemble Plants homepage, you've got the answer is that you need to go to the FTP site. So what you can do is you can go to this link here at the top that says downloads, and you can actually find information about the FTP site, and you can go there directly if you want to, and you can begin exploring the directory using the release number. And then you said you wanted a GTF file, so you can click on GTF. And then if you want your species, maybe Arabidopsis, you can find the GTF file there. If you want to be a bit more direct, you can come back to this page that we had by clicking on downloads from the Ensemble Plants homepage. And you can actually find this table that shows you all of the different files for all the different species that we have. So if you can search for a particular species, maybe you want, Arabidopsis. We've got 3 different Arabidopsis species, so Thaliana, Haleri, and Lyrata. And you can see again, we've got obviously the GTF and the GFF3 file gene sets here, which you can click on. and that will take you out to the correct directory in the FTP site for you. So Sweeta has got back. The GBS stood for genotyping by sequencing data. So I'm not sure what file format that you have there, but I don't think it's I don't think you can submit your GBS data set to Ensemble for analysis. I think you'll need to look into other platforms that you can use to analyze that data. And then Dr. Sarva Mangala says, if we are loading our BAM file that is not publicly available yet, research can access my BAM file. So no, the answer is no. So if you upload your data to Ensemble to visualize in the Ensemble Genome Browser, like we saw, here with the custom tracks option, that's just a way to visualize your own data set. So this is not a way for other people to access your data. It just means that you are looking at your own data set in the context of the Ensemble data. So what you can do is you can create an account in the top right hand corner here by clicking login/register. And what that means is that it just creates like a workspace where you can save your data file, and it means that you can just visualize your own data alongside the publicly available data. So someone else coming to Ensemble won't see your data or be able to visualize it. Rishika has asked, can I download a CDS file also from Ensemble? And the answer again is yes. So just as we saw before, if we go to the Ensemble Plants homepage and click on download, you can see that one of the sequence files that we have available is CDS. So we've got DNA, cDNA, CDS, and as well as protein sequences as well. So if you want to download the FastA CDS, the CDS FastA file for Arabidopsis thaliana, then you can click here, and it will take you to the correct directory on the FTP site. Now, we've talked about the FTP site quite a lot. It's important to remember that these files are genome-wide files. So the GTF file and the CDS FastA file, these are going to contain information about all of the genes across the whole genome for that particular species. So if you want a whole genome file, then that's obviously this is the place to come. But if you just want to get CDS sequences for a small set of genes in a particular region, then there are other tools that you can use as well. But I think this is exactly what you want. Okay, so I think we've come to the end of the questions that came in. So we just have around just over 15 minutes left together. So what I would like you to do is spend some time having a go at the exercises. So if you go back to the training materials, and I can paste the link back in the chat box just so you can find it again. I'd like you to have a go at the section here that's called Species and Genome Assemblies exercises, and then also exploring genomic regions exercises as well. Again, you've got the answers there, so please do have some time. Please do spend some time to look at those if you get stuck. But Alina and I will be here to answer any questions that you have as well. So if there are no further questions, then please do have a go at those exercises. Alina and I will be here for another 16 minutes until 8:30, I think it is in Indian time. And then just before that time is up, I will come back and see how you've got on, and then we'll be ready for Alina's session tomorrow. Hi, everyone. This is just Ben here. We're a few minutes to 8 o'clock, so it's nearly the end of the, sorry, half past 8 there in India. So we're nearly at the end of the session today. So I've been keeping an eye on the living document and the chat box, and I haven't seen any further questions come in. So I hope that you've been able to have a go at the different exercises. I just wanted to give you an update for the plan for tomorrow. So as I mentioned, I'm joined by my colleague Alina in the Zoom call today. She's been answering some of the questions in the chat box. Alina is going to be presenting the genes and transcripts module tomorrow. So what Alina is going to be thinking about is how the gene annotation process works, so how Ensemble actually annotates the genes that you are visualizing in the Ensemble Genome browser, and how we can get more information about the genes and their transcripts. So it's the same time again tomorrow. So it's 7 p.m. I think Indian Standard Time for the session tomorrow. So thank you very much for joining us, and we'll see you tomorrow.
So thanks, Ben, for such a wonderful session on Ensemble Genome Browsers. And today tomorrow we will meet again with the Ensemble outreach team for another session on Ensemble Genome Browser. So with these words, now I would like to thank Ben for his session and seek his permission for ending today's session.
No problem. Thank you very much, and it's nice to meet you all. Thank you. Have a nice evening.
Thank you
very much. Bye.
Bye-bye.
Bye. Thank you.